Well, I should say, I wouldn't say the biggest, but it's one of the grand challenges for our generation because, we don't understand completely how our mind works. We don't, and there are significant problems in society today related to that, such as depression, autism, addiction. They're all related in some way to the brain. And so I think there have been some bold programs like to map the brain, to create an atlas of the brain, because if we can understand how our brain works, we can first of all address many of these big issues, and also we can start to ask very profound philosophical questions like what is consciousness and what is pain and what is the meaning of intelligence.
Welcome to the Navigating Nano podcast, where we explore the multidisciplinary world of nanotechnology with distinguished professors and researchers. Join us as we sit down and talk about their cutting-edge research, career paths, and expert opinions on the expanding world of nanotechnology. Dr. David Gracias joins us this week. Dr. Gracias is a professor at Johns Hopkins Chemical and Biomolecular Engineering Department, known for the design and development of miniaturized devices. He sits down with us, the Materials Research Society at the Joint School of Nanoscience and Nanoengineering, to talk about the benefits of reading literature outside scientific papers, networking, and what can be solved if science could just crack the code to debatably its toughest matchup, the human brain. Join us as we navigate Nano.
Hi everyone, welcome to Navigating Nano. I am your host Anjali, and today with me I have my co-host Isaac and Dr. David Gracias. So in today's session we'll just be asking him some questions about his journey and himself, and we're ready to get started whenever you guys are ready. All right, so a little bit about him first. So Dr. David Gracias is a professor of chemical and biomolecular engineering at the Johns Hopkins University. He's a pioneer and leader in smart miniature devices, and he has done many collaborations to put his inventions into practice. And right now he currently holds 33 patents, and we're very delighted to welcome you at the Joint School of Nanoscience and Nanoengineering. So welcome.
Welcome. Thank you.
All right. So to get this started, first off, this is more of a technical of what we know about you, but tell us a little bit more about your journey, who you are, things you like, academics, personal, anything that you want to share with us.
Yeah, so I grew up in India. My name is, it sounds Spanish, but I'm from Goa. It used to be a Portuguese colony, and so the name is inspired by there. And I did my undergraduate there, and at around 21 years old, I came to the U.S. For my PhD, which I did at the University of California at Berkeley. I remember the first flight to the U.S. was in San Francisco, a lovely airport, and I still remember it. And I worked at the University of California at Berkeley. It's a great institution. Many people don't know, but in chemistry, it's one of the powerhouses in chemistry. Several chemical elements were discovered there, about 10, I think. And there's even an element called berkelium. So when my friends, we have rivalry with Stanford, and when they used to say, well, Stanford is better, we would say, well, is there an element called Stanfordium? I haven't heard that. And so, and then I did a postdoctoral fellowship at Harvard on the East Coast. And I've worked for a couple of years in industry at Intel, the chip manufacturer. And then since 2003, I've been at Johns Hopkins for almost 19 years now. And I run my own lab, yeah.
Nice. So just a little bit follow up on that. So what was it like working in all of these bigger schools, bigger organizations? Did you ever feel intimidated? Like, how did you find your place in them?
I did feel it was a bit overwhelming. I remember at Berkeley there were many Nobel Prize winners in chemistry. Sometimes you would be at a seminar, there would be a Nobel Prize winner sitting next to you. There were, Berkeley has a very tough parking situation, and so the ultimate joke on campus is that if you become very famous, you get your own parking spot. So, like, there were parking spots for like Stephen Smale and others, who are very well renowned, even beyond Nobel Prize. So there's like Nobel Prize reserved parking spots, and then there are parking spots with your name. So it was, but also it was, you saw people as humans. So there was, the Nobel Prize winner was made of the same flesh and often could make mistakes, was very human. So it, I think hanging around very accomplished people, you see that there's not, you can also be, they're not like aliens or something. Like, they're made just like you. And Harvard was also a great experience, a very flourishing.
For like new students coming in and like, being intimidated by like bumping shoulders with a Nobel Prize winner, right? Like, for you, what was the most scared experience where you were like, oh my God, I'm a fan. Like, I'm such a fan, but like, I don't know how to like approach this moment.
Yeah, so I think the natural thing is to, the first thing is to be scared to speak up and all. But over time, you lose those inhibitions and you start to...
Well, was there ever a moment where you like met somebody and you were like, oh my God, like this person is like it?
Yeah, many times. I think one of the highlights is like Charles Towns. He's the inventor of the laser. And I remember he gave a talk, and it was very intimidating at the end. And he was actually talking about how he came up with the idea for a laser. And at the end, I remember asking him a question and feeling really, is the inventor of the laser.
What can you ask him?
Yeah, exactly. Yeah.
Right.
So,
so you're also a mentor.
Yeah.
You have, I saw a big list of mentees. Like, when you're interacting with them, like, what are you trying to instill in them? What are you trying to like get them to really grasp when you're trying to inspire or whatever?
Yeah, so mentoring, I've been a professor for 19 years, and I've, been through different phases of mentoring. I think I also have a 9-year-old son, so I think after I had a child, I also changed a little bit as a mentor, because I saw the process of learning. In humans, it involves like trial and error,
right?
Making a lot of mistakes. So when I saw my son walk, he would get up and fall, get up and fall, get up and fall. And I actually was like, in my mind counting, it's probably thousands of times. And I think this is very important for students to remember also, that failure is part of the learning experience. Like, if he had, I was just telling the other students, if he had got up and fallen and said, well, I'm now not going to do this again. This is not for me. I'm not able to walk. He would not have been able to walk today. So somehow when we are young, we are okay with like failing and all, but when we get older, we become more conscious. So I think when you're a mentor, you have to have patience, and that's, and I found also that, each human has different skill set and different. And I think the challenge of a mentor is not to push the mentee into a particular whole shape, but to bring out the best in what they can do. And that's always a challenge, because you wanna think that there's only one way to be a great scientist, and the natural tendency is to push them to that way, which but actually there could be many ways to.
Right, because you have your own experience that you were like, I took this path.
Yes,
this worked for
doesn't
me.
mean it's for everyone.
Exactly. That's awesome. What was your favorite project to work on early on that inspired you to keep going? Because it's really hard, like you said, trial and error constantly. That's the life of a researcher. Sometimes it's can I keep going? What was a project that kind of inspired you to just keep going? And you were like, I can see the magic happening.
So I think there are, three types of research, and I've been fortunate to experience all of them. One is the research of discovery. And, when I was a student at Berkeley, my lab, we typically spent several years building an instrument, which was very hard to build. And then we would do some measurement, so that was very hard work, actually getting something to work down to the last level, the noise, and also that you can do a good measurement.
Yeah.
So I like that part, but during my postdoc, I worked on more creative things, like inventing things, and I think I've really molded my career more after that. And that was shocking. I worked on this paper which was published in Science magazine on this idea of, can we build a computer by just throwing things together? Okay, so it's a very radical idea. It's the idea of self-assembly, which really hasn't caught on in engineering, but it's this idea of, in human engineering, we are always about cutting things or building things. And in nature, if you ask how is a virus made? It's made by proteins that come together on their own. Mm-hmm. And they lock in place, and the forces are perfect.
Right.
And so my postdoc advisor is lab, and he's a very influential George Whitesides. We had this idea of, can we build a computer circuit this way? Okay, and we had blocks, and then we had to figure out the patterns so that they could stick in particular ways, and we pulled it off. I think it's a very impressive paper. It's one of my most beautiful pieces of work. And also it showed me the third area. So I think in research there are three areas. One is discovery, one is analysis, where someone gives you a bunch of data and then you make sense out of it. That I would call analysis. And the third is invention, which is you create stuff out of nothing.
Right.
So, you make a computer circuit that, And so, mostly now I like creating,
Inventing.
Yeah, I think of myself also as an artist.
Yeah.
But I know the tools of physics. My tools are physics,
right.
Chemistry, and I'm trying to create a painting with tools but very rigorous tools.
Still scientific, still using that logic and that intellect.
Yeah. And I found there are many pressing problems in society that engineers and scientists can address. And I feel we all need to. One of the things that is a bit disheartening to me is that people don't think of scientists as cool things. There was nerds and this and also like this discourages some of the smartest kids. They don't do science, engineering or science. They go into like law or medicine or something, which are also great fields, but they are considered like cooler. So I think the joy of discovery and the joy of creating things is unparalleled. Like I cannot imagine myself doing anything else at this point. Like I get so much joy out of discovery and create.
When you're inventing, creating, that's like I feel like that's the experience of being human, right? Like that's what you want to get to as a researcher, and I feel like what is hard, especially when you're just starting out, is you have no ground to work with, right? You're just kind of out there floating, and you're like waiting for something to grab onto that you can be like, I started inventing something. So what do you think is like the key to like getting those brilliant minds out there to like latch on and like start to become inventors and not just like researchers?
I think, like humans, like my son learned to walk, you really have to do it once, see that you can do it, and then you begin to think that it's not so hard to do that. And it's just like learning. I think there are two barriers to two very important things that, scientists may not appreciate when they're starting off in research or in invention. And that is, one is that failure is very common. And people don't deal with failure well. Like walking, you have to just, if your experiments fail, your advisor's not happy, whatever, it's natural. When you're dealing with the unknown, you're going to fail. It's like an explorer. You cannot just go and get and if you want to read about great scientists, read about their failure. Like I was just reading the Darwin's book, The Voyage of the Beagle, and it's full of failure. Actually, it's just, you just hear like people are dying on his boat. They are like getting shot. I was thinking like, my gosh, if I was a scientist, I would get off this trip. Like, this is serious stuff. Yeah. Like, people are dying out of disease, and some are not making it to the place. And we just think he went to the Galapagos and he just saw some finch and he's made a theory. But if you read the details, and this book is very long, and it stops and he gets sick, and and so failure is very common. If you cannot handle failure, I think it's not going to be. And the second thing is it's a bit slow, and I find like many sharp kids, they want like instant gratification. Like if you're a stock trader, you might place a bet in 3 seconds. You might know if your bet paid off. Like if the stock went up, you made millions, and otherwise you lost. But in research, if you take a gamble, like I'm going to make a self-assembling circuit, it might take you 2 years to figure out whether it didn't work out, right?
Yeah. And if it didn't work out after 2 years, you're like, well, that was 2 years of my life.
Yeah.
So kind of building off of that, honestly, so I want to go back to your presentation from today, because I know you mentioned a lot about discovery and invention, but at the same time you said that the biggest challenge, especially for our generation, will be to understand the brain. So why did you specifically mention the brain, and like what was your reasoning behind it? Because you were talking about like all of these gels that you're innovating, but why the brain?
Well, I should say, I wouldn't say the biggest, but it's one of the grand challenges for our generation, because we don't understand completely how our mind works. We don't, and there are significant problems in society today related to that, such as depression, autism, addiction. They're all related in some way to the brain. And so I think there have been some bold programs like to map the brain, to create an atlas of the brain, because if we can understand how our brain works, we can, first of all, address many of these big issues, and also we can start to ask very profound philosophical questions like what is consciousness and what is pain and what is the meaning of intelligence. And so it's even important from a computer science point of view as machines start to get more human, like AI and neural networks and all. So the computer scientists are also asking, what is really human? How does a human drive a car? I mean, after so many years, they're trying to build self-driving cars, and you have a 16-year-old, they take a test, they'll take 3 weeks of lessons, and they learn to drive a car. And so there's something special about the way we perceive space, we perceive images, that we can drive, with a few weeks of practice and computers and all that, their facial recognition, music, what does music and sound interface. And so it's important from the computers, it's important from disease, and it's important from philosophy, just who we are, what we are. So I think it's one of the frontiers of the future.
Do you work with like psychologists or like other like fields of research within Johns Hopkins to kind of like, because the brain, I mean, there's so many ways to study it. So, approaching it from a multidisciplinary standpoint might be the most productive. What do you think?
Yeah, we have I cannot, I mean, it's too big a problem for one person to, or even one university to look at. But we have a huge number of people at Hopkins who are all the way from psychology, philosophy, ethics, medicine. And I don't collaborate, I wouldn't say, but we are sometimes in consortia, and we recently had a workshop. And so, and whenever I can, I try to read books on philosophy and psychology. My wife always makes fun of me because I don't read any fiction books. I only read non-fiction, and she only reads fiction. So it's interesting. But I try to read. So I told you Oliver Sacks, who I presented. I've read most of his books on those books itself are. Some of them are too shocking to even believe, okay? Like, he has a book on hallucinations, like how the brain perceives image, and some of the experiments that have been done, which are like very shocking, like they put what happens when you deprive stimulation from humans. So I was shocked that the literature on this is very profound. Like they've put people in like experimental studies where they put people in like a box. Okay, you're sitting comfortably, but there's no stimulation. It's a soundproof box with like black thing, and there's plenty of air. These are study people, and the brain sometimes will create hallucinations and create, So even the blindfolded, the sensory deprivation and things will. So some of his books are, because they are again all clinical. He was a clinician, so he talks like all studies on humans. It's a bit. It asks questions like what is vision, what is like he talks about some patients who cannot see 3D, and there are other cases also like that. They can only like see 2D and things like that. So it's the human brain is quite amazing. It's almost like reconstructing the world around you.
Yeah, that's very fascinating. Yeah. But enough of us asking questions. I'll open it up to our audience to ask some questions. So whoever is ready can go first.
I have a question about your research. So you talked this morning about tubes being able to grow with infants, like tubes in the heart. I was wondering what kind of environmental changes would cause that growth. because I would imagine like inside the body is pretty stable. So I was just wondering about that.
That's a great question. So it comes back to the one of the challenges, as I mentioned, is to have implants that adapt to the body. And in this case, you want implants to grow when there's a surgery in a neonatal or very small baby. As the baby grows, you would like the tubes, artificial grafts, they call them, to grow. So that's a great question you raise, what stimuli. So we've thought about it in two ways. So one is that there are other changes that occur in the baby, like flow rates can change so the pressure and as you grow, the dimensions change. So there are other triggers that are changing with the baby. So for example, if you had a tube that will grow with flow, you can also have something that is timed. So if it has multiple folds like an accordion, maybe the material just starts to elongate after a while just because of what, it could be like creep, kind of a creep-like phenomenon. There are other ways we've thought of which are like surgical interventions, like you have some magnetic particles or something, and then the baby, when they're like older, they go to the hospital, but they don't have to do surgery. They can just like get an MR or some external power applied to move one joint. So that will be much less invasive than a surgery, if acoustic or so. If you have something that can be elongated with a trigger, external trigger. So that's the second way, and that if that couples with temperature, it could couple with other conditions. But it's a hard problem because in our case, the tubes we were looking at, they connect directly to the heart, and they have blood in them. And whenever you have blood, the cardiovascular system is very sensitive to the material, because you can get clotting, you can get foreign body response. So it's a hard problem. But I think as inventors and discoverers, we need to just think of first simple as an engineer. Can we even make something that we can grow? Then we can also think about the material requirements. And there was a question about immune and other things. And these problems already occur with normal grafts, so people already worry about this. Many grafts fail because of accumulation of debris and clotting and other things. Yeah.
Thank you.
My question is about hydrogen. I'm wondering, how did you control the swelling degree of your hydrogen? And the second question is about the zeta potential. What was the zeta potential of your hydrogen?
We haven't measured the zeta potential. So we have two kinds of hydrogels. One is the temperature responsive. And you basically can control the degree of swelling by the amount of cross-linking and the size of the polymer chain. So it's like a net, a fishing net. So it's very intuitive. If you make the nodes in the fishing net longer, or shorter, you can change the degree of cross-linking. So that's one. We also can co-polymerize. So we can put different other polymers in. Yes, yeah. And the DNA gels, DNA has charge on it, so yes, the zeta, but we found that the degree of cross-linking is based on, we use what is called strand displacement. And so the length of the strands, and in our paper we published a semi-empirical, so we have found relationships that we're still trying to understand from a theoretical standpoint. It's a new kind of material. The reason it's a bit complicated is there's charge involved. It could be like osmosis. the water coming in. But there's also elastic strain because DNA is coming into the picture. And we're still working out all those details. Yeah.
But have you been able to see the interaction between the gels and the DNA for you to conclude that there's that interaction?
In what do you mean by?
From an experimental point of view. So have you done it experimentally to see the gels interacting with the?
Yeah. Well, we can see that we can fluorescently tag the DNA. We can see them entering the gels. And we can also see the gels swelling with different... We basically do a macroscopic swelling experiment. So we build a block like 1 millimeter by 1 millimeter, many of them. We can put them in dishes and we can add the DNA and see literally with a microscope, measure the final size of them.
Yeah, I'm asking that because I think in tissue engineering, a lot of people use this stimuli responsive kind of gels. So I don't know if they are aware of that reaction between the gels and the DNA. It's very conflicting knowing that there are people already using them, and yet there's this challenge of that interaction.
No, our gels we are using for robotics and What I didn't understand, I'm not completely understanding your question.
Okay, so
yeah.
There's some people use these gels for polymers.
Yeah.
For tissue engineering.
Yeah.
And they are transplanted in people. So that would mean the long-term effect of using these gels on real human samples is the degradation of the gel over time.
Yeah.
So could that be your advice to the people already using these gels because they have this challenge? What would that be like? Should they stop using it because it is a progressive field already?
No. The mostly the DNA gels are not used in tissue engineering. They use very simple gels like alginate and polyethylene glycol diacrylate. Those are gels. Silicone-based gels, things like that are used in gelatin, collagen. These are used in tissue engineering. Yeah. Our body has gels, right? So we are made of collagen, a lot of which is a gelatinous material. And in tissue engineering, the challenge is rejection of the tissue. So you can put cells in a gel and you can implant them. But first of all, you can elicit an immune reaction, so some of the materials they use in tissue engineering, like Matrigel and other things, can come from animals. And you can get, may have contamination or may, and so the challenge is always synthetic versus natural polymers. And but people who are using them, they test them. So they put them in the best way to know if something works is to test it, right? And if there's an inflammation, you can see the inflammation if there's an immune reaction. And if there's rejection, you can see rejection. And yeah, sure.
And did you have any activation or functionalization aspect in your fabrication of hydrogel? My work is in hydrogel, and because of that, I'm really interested to know about it.
Yes, we have done chemistry on them, but very simple chemistry, not too long, just like adding side chains or polymerization units like acridite and other kinds of double bonds in there. But we are not, I wouldn't say we are synthetic chemists, so we do very simple. I did have a chemist in my lab who made a NIPAM gel by changing and poegma gels by changing the length of the crosslink to make, we had a paper where we had this gel, 3 gels that activate at different temperatures. So one is like 20 degrees, one is 27, one is 32, and we wanted to make like a multi-state device. So as the temperature increases, they do different things. But elsewhere, there are many researchers who focus on making gels of different chemistry. Yeah. And the DNA gels, the beauty of DNA is the programmability. It's a very Watson-Crick programmability is very efficient. So we are using that and basically strand displacement to, yeah.
And regarding these DNA-based hydrogen, is there any software that we can design the primer, for example, in rolling circle procedure for entanglement DNA-based hydrogen, to design the appropriate intervals between 2 or 3 primers, which at the end have the DNA-based hydrogen with the appropriate pore size? Is there any software? Or we should do it manually?
So there are a lot of software for DNA nanotechnology. So how they bind, there's one out of Caltech, the Winfrey group, and others, Rothermund, and then there's, software from Europe, etc. Our gels, so there's 2 kinds of DNA gels. So one is DNA gels that just bind DNA to DNA. Okay, and those were the conventional gels before our paper. Our paper uses like a polymerization network. So its hairpins keep coming. And there are also termination hairpins. So to my knowledge, there is no, readily available software. But, if you would like to know more, you should reach out to my collaborator, Rebecca Schulman. She's she has a computer science background and she's from the DNA field. And, she designs the circuits. My role in the project, and when we co-invented this, there were all these circuits, but there was no material. So my role was to, first of all, find a way to put this in a gel. Okay, there were all these circuits binding and all. We wanted to make a macroscopic material. So if you know, DNA nanotechnology is an old field. But most of the structures people build are 100 nanometers, 20 nanometers. But our focus when we worked on this work is this concept of digital material. So we want to make a material that has little parts, each of which we can swell or shrink. Okay, so that requires a lot of. First of all, you need a gel. We help take the circuits with Rebecca and cross-link them to make gels. Then we learn to process the gels, okay? This is not easy because DNA is expensive. We don't get gallons of it, okay? We buy the DNA and we get a little vial and we have to do microfabrication with a little vial. So there's a lot of technology I didn't discuss, which is we make these little chambers to pattern them. We have to put the DNA to make that voxylated material. We need to put different gels at different spots. Okay, you can't 3D print these because you can't make an ink. So we photo pattern them. We mix them with cross-linkers, and So really, Yeah.
May I know your collaborators?
Sorry?
May I know that collaborator that you mentioned? I can.
Yeah, Rebecca Schulman. She's one of in my department, a very brilliant young professor, and yeah.
Thank you.
I have a question for your drug delivery device. So
Yeah.
you have mentioned that you used gold device or a device made of gold. And we know that usually gold, if you go down with the size, they become reactive. So have you tested the stability of your device with biological samples? I mean, when they interact with the proteins, they start to form protein layers around them. Are still doing the drug delivery, or what is the stability of the particles in biological samples?
So there's a lot of history of using gold in the body. I have actually a gold filling in a gold cap. But your points are well raised. Safety, we plan to work. So I work with clinicians who actually work with patients, right? So my collaborator is a MD who actually sees patients. So they keep us very real, okay, because they actually know what works in the clinic. So at the thin film length, we are talking about gold is not very reactive. Maybe at the atomic scale it might be, but we had the 100 nanometer length scale films. We don't think it's reactive. And as I mentioned to you, we are using it in the gastrointestinal tract. The gastrointestinal tract is renewed every 4 to 7 days. So the whole lining goes off. So if there are things stuck on it, they also go off. And over those 4 to 7 days, we can only say what we have done, which is, I told you, we did safety studies in pigs where we put them in and we did not see any trauma or anything over the course of a few days. But definitely to get FDA cleared and all, there will be more rigorous studies and more extensive studies that will need to be done. But they will come about only when we have to demonstrate very clearly that we can give insulin at the right dose. And, we are trying to develop ways to deliver the injectors, etc. So I would say it's still relatively early stage. And we need to work on that.
And how was the release of the device? Was it like a sustainable release, or was it like burst release?
Yeah, so we piggyback on the drug delivery community. So we put a patch. Our device is just a simple machine that closes. Okay. What we put on the back is, you tell us what to put, we'll put it. Okay. So it's just a simple machine to inject. Now we have used chitosan, and chitosan is a well-known drug patch, and but we are exploring other things. We are also exploring reservoirs, like little holes. So we put a free drug, things like that.
Yeah. And what are advices you want to give for new researchers, to move on from the small hood we are working on to real life?
Yeah, I think, it's really a personal thing. I think the nice thing about a PhD degree, are you a PhD student?
Yes.
Is that it's a very broad degree. Sometimes I compare it to like a law degree, okay? You can use it for many things. You can use it for research. You can go into, policy. You can go into law. You can go into business. You can go into, and over 20 years, I've seen my students, they've gone in all different directions. And they've all been successful. So the question is really, what do you want to do? And that's a personal decision. So I think you have to find out what your strengths are. I found it's very good to talk to people also, and who are already doing these jobs, and ask them what does your daily life look like? What time do you get up? Things like that, okay? And, do you have any geographical restrictions? Do you have money restrictions?
Yeah, I also have a question about the microfabrication. So, you mentioned that you use aluminum and instead of silicon. So my question is, what advantages do you find with using aluminum over silicon? Why did you choose it?
We use germanium as a sacrificial layer to release the structure. Germanium is water soluble, and germanium oxide, based on the stoichiometry And this is quite tricky. Depending on the way you deposit germanium, you can get different oxide states. And they all dissolve at different rates. So you can actually time it to dissolve at different times, and we have this know-how in our lab, so we can, for some applications, we want to dissolve it in few minutes, otherwise in few hours, things like that. So, yeah.
Another question now is about the organoid media that you mentioned. So we use a lot of media here, and the conditions are quite 37 degrees, 5% CO2. Does that apply for the same the organoid media?
Yeah, we need media conditions, and yes.
Yeah, so this organoid media, I was just curious.
Yeah.
Does it grow the organ? Because it is different. You mentioned that it's very different from the media that we use, but I didn't understand the concept of organoid media over this other culture that we use.
So what is media, right? So media is basically like a cocktail of chemicals, okay? And different chemicals are needed for different stages of the organoid. So while it is growing, you need some particular media, and then when it's grown completely. So we actually work with a collaborator. Again, she grows the organoids. There are well-published protocols. So if you go to Aldrich and you buy organoid media, it will tell you exactly what to do, like when to put it and what to put. And it's like a recipe, cooking recipe. So you follow the recipe, you get your steak well done. There's and then once it's grown, because it's not vascularized. So this is a challenge in the organoid community. One, we are working with a collaborator too. We're trying to put microfluidics, because once you get to a particular size, like 0.5 millimeter, you start to get a necrotic core in the center, right? Because there's not enough diffusion to the center, and you can't grow it very big unless you have fluidic channels. So two challenges that if you are interested in the community of microfluidics, one is to grow the organoid on chip. Right now, they don't do that. They make it in flasks because you have to shake a lot. And then, so it's quite hard because we get the flask, we get the dish with, I showed you those white dots. And then the student has to move them to the chip, which already can damage the organoid. So ideally, you would, if I had a magic wand, I always start with the magic wand. What is the ideal condition? The ideal condition is that we build, and we've been trying to do this for many years, a micro incubator where we can shake, do everything on the dish, on chip. We put some cells there, we make the organoid on the dish. We have all the micro instrumentation for the organoid. And so I think in cell culture, again, my expertise is in microtechnology, so these are the challenges. How do we make instrumentation for small things? Microfluidics is great, but conventional microfluidics has some limitations, like it's mostly two-dimensional. It's the channels are mostly like square cross-section, which is not like the real body. And it's hard to make crossings and very complicated structures.
All right, so I think that's all we have time for. We can take questions later after the series, but thank you so much, Dr. David, for coming with us today, and we'll see you next time at our next session of Navigating Nano.
Okay,
thank you.
Thank you.
Thank you so much.
Are you a student?
Yes.
Yes.
Yeah.
All right, Dr. David, before you leave, I have a quick form for you.
Yeah.
Yeah, it's just so that we can kind of use.
Thank you for tuning to the Navigating Nano podcast. Special shout out to Dr. David Gracias and our host for this week, Anjali and Isaac. Be sure to follow us on our other socials, especially on LinkedIn, YouTube, Facebook, and Instagram. Thank you. See you next week.